Now we're going to take a look at the different types of intermediate filaments. The structure of all intermediate filaments are similar in that they share these features. There's an alpha helical rod that binds with another to form a coiled-coil dimer. The dimer then binds with another dimer to form a tetramer. And the tetramers then bind together to create a protofilament. Different protofilaments then bind together to create a proper intermediate filament. And as we already know, some of these intermediate filaments can attach to organelles to keep them in place. Now let's take a closer look at how these dimers bind together. At the end of these helical structures, there are protein chains called head and tail structures that can bind together to glue these dimers together in a rope-like structure. Most of the intermediate filaments are made up of homodimers, meaning that the dimers are made up of the same alpha helical rods. But keratin filaments, also known as type 1 and type 2 intermediate filaments, are made up of heterodimers that then, just like every intermediate filaments, form tetramers and then protofilaments. And keratin filaments can, for example, be found in the keratocytes within the skin. And in these keratocytes, they serve the purpose of attaching to each other and giving a tensile resistance force towards stress. Now let's take a look at type 3 filaments. These filaments are further divided into 4 subgroups, and we'll take a quick look at where each of these filaments are found. The first one we're looking at is glial fibrillary acidic protein that are found in the glial cells in the brain. Next up is vimentin, which are found in neuroblasts. Then we have desmin, which are found in smooth muscle cells. And lastly, peripherin, which are found in neurons. Now moving on to type 4 filaments. They are also mostly found in neural cells. Then we have type 5 filaments, which we found, for example, within the nuclear membranes of the cell core. Lastly, we have type 6 filaments, and the only protein found so far belonging to this group is called nestin, and they are found within early developing neurons. And before we finish this video, we're going to take a look at how remodeling of intermediate filaments are done. Intermediate filaments don't undergo dynamic remodeling like the other cytoskeletons. Instead, within the nuclear membranes, for example, when the cell needs to divide, there are these phosphorylizing proteins, shown in red, that phosphorylate the head-to-tail region of the intermediate filaments, causing them to split up, and thereby also destroying the nuclear membranes. This is important during cell division, where the chromosomes needs to be available for separation. And that covers everything for this video. I hope it was helpful.